Okay guys, in the previous video we were discussing about G proteins and we have so far discussed about G stimulatory proteins, and in this video we'll discuss about G inhibitory protein. G inhibitory protein and their receptors are also present in the similar manner as the G stimulatory protein. So it will be present outside the cell. This is the ligand binding part, and since it's a serpentine receptor or the 7-pass receptor, it will enter 7 times. And here it enters, and it is coupled with G inhibitory protein, so it will be alpha inhibitory, beta inhibitory and gamma inhibitory part. So this is G inhibitory protein, and this was G stimulatory. So all the process are almost similar. During its inactive stage, it is bound with GDP. When any ligand comes, for example, acetylcholine, that is muscarinic type. It will activate the receptor will send signal to the G proteins, and G protein will take up GTP. GTP will come and GDP will be kicked out, and the alpha subunit will dissociate. It will be the alpha inhibitory unit with GTP, and the beta and gamma subunit will also get dissociated out together, beta inhibitory and gamma inhibitory. Now in adenylyl cyclase there is a receptor for alpha inhibitory side, for the alpha inhibitory protein. So it gets bound to this receptor, and what it does, it inactivates adenylyl cyclase. Adenylyl cyclase, when gets inactivated, it reduces the CAMP level, and this in turn will not lead to activation of the inactive protein kinase A. So all this process will not occur, and the cell will get restored to its previous value. So now we will discuss about another type of G protein coupled receptors, and these are the GQ receptors, and they are basically found in the smooth muscle cells. And GQ receptors are quite different from the G stimulatory and the G inhibitory receptors in terms of their action. So let's see. These are also a class of serpentine receptors, or the 7-pass receptors. It has an extracellular domain and an intracellular domain. So let's take an example of oxytocin or epinephrine. If we take oxytocin, it will be better. So if oxytocin stimulates the receptor, the receptor will undergo certain changes, and it will in turn stimulate the G protein that is associated with the receptor. This G protein has 3 subunits: the alpha Q, beta Q and the gamma Q subunits. Unlike other G proteins, in its inactive state, this G protein is associated, or rather the alpha part of the protein is associated with GDP, and when receptor gives signals to it, it throws away the GTP and takes the GTP unit and gets activated, and it gets detached from its beta and gamma subunits, and it moves towards another protein that is present in the cell membrane, and this protein is known as phospholipase C. It activates, or the alpha Q subunit of the G protein activates phospholipase C. Phospholipase C is very notorious, and it breaks down its own component of the cell membrane, and that component is known as PIP2, or phosphatidyl inositol diphosphate. And on breaking PIP2, the PIP2 further is divided into 2 parts after its breakdown, that is IP3 and DAG. IP3 is inositol triphosphate and DAG is diacylglycerol. Now let's move to Now let's talk about each of the components of this PIP2. IP3 or inositol triphosphate, it activates certain pumps. Those are present in the endoplasmic reticulum, or they may be sarcoplasmic reticulum in muscles, and these pump actively pumps out calcium ions from the endoplasmic reticulum or the sarcoplasmic reticulum into the cytosol. And calcium has various roles. It can bind to troponin, it can bind to a protein which is known as calmodulin. On binding with troponin, calcium exposes the mask sites of attachment of myosin with the actin filament. So it brings about contraction of the muscles. And if it binds with calmodulin, calmodulin in turns activate calcium calmodulin protein kinase. This is a type of protein kinase, and this calmodulin protein kinase can phosphorylate enzymes, it can phosphorylate transcription factors, it can phosphorylate ion channels, and like other protein kinases, it has various roles. And all these things, phosphorylated enzyme, phosphorylated transcription factor, phosphorylated ion channel, can bring about certain metabolic changes in the cell. And another component of PIP2 is DAG or diacylglycerol. DAG can activate protein kinase C. Why we will discuss about protein kinase C? Because it is associated with phospholipase C. So activated protein kinase C activates certain enzymes. It can phosphorylate transcriptional factors. It can phosphorylate ion channels, and so on. It can bring about so many types of metabolic changes in the cell. So this was all about GQ receptors. You can take a screenshot of this and write it down later. It is the GQ receptors. Okay, now we'll discuss about another aspect of receptors, and I think so you have heard about cholinergic receptors, adrenergic receptors, dopaminergic receptors. So let me give you a brief idea about all these things. Cholinergic receptors are of two types. They are nicotinic receptors and muscarinic receptors. Nicotinic receptors are ion channel, and remember that these are not G protein coupled receptors, but the muscarinic receptors are serpentine receptor, or they are also G protein coupled receptors, that is they are the 7-pass receptors, and they are acetylcholine mediated. That's why they are known as cholinergic receptors. And there are certain receptors which are adrenergic receptors, and adrenergic receptors have many subclasses, like they are alpha 1 category of adrenergic receptor, alpha 2, beta 1, beta 2, and beta 3. Each have different properties or different G protein associated with them. The alpha 1 adrenergic receptors are associated with GQ proteins, and you have seen previously that GQ proteins finally break down PIP2 into IP3 and DAG and give certain metabolic actions. Alpha 2 are GI, that is G inhibitory protein, and they decreases the cyclic AMP level, as in part 1 I have showed you. And the beta and gamma inhibitory part, they helps or they readily removes potassium ions from the cells. Beta 1, beta 2, and beta 3, all these are G stimulatory protein, and they increases the cyclic AMP level, and finally protein kinase A level gets rise. And there is another class of receptors, those are dopaminergic receptors. Dopaminergic receptors are of two types, D1 and D2. D1 receptors are associated with G stimulatory protein, and D2 receptors are associated with G inhibitory protein. And you know that G stimulatory protein can increase the cyclic AMP level, and G inhibitory can decrease the cyclic AMP level. So this was all about the cholinergic receptors, adrenergic receptors, and dopaminergic receptors. Okay, so now we will discuss about intracellular receptors. These receptors are present inside the cell. Previously you have seen the many extracellular receptors. They are present outside the cell membrane, and these receptors, which are the intracellular receptors, are present inside the cell membrane. And which substance will have the receptors inside the cell, those substances that can easily or readily pass through the membrane, like steroid T3 and T4, that is of thyroid hormones. Vitamin D, it is also a type of steroid hormone. So remember, STD. All these all these categories of hormones can enter inside the cell membrane, and their receptors are present inside the cell. So these are the intracellular receptor. Now let's see how they work. This is the ligand, or this is the hormone. Anything, it can be anything, steroids, thyroids, or vitamin D. It can easily pass inside the cell, and the red color thing that you are seeing over here, this is the receptor or the intracellular receptor. When there is no hormone, or when there is no ligand, the DNA binding side of the receptor is covered by a protein. This protein is known as heat shock protein. This protein covered, or this masks the DNA binding domain of the receptor. When the ligand approaches it, the receptor gets activated, and what happens, the heat shock protein is removed from the receptor. See, when the receptor gets associated with the ligand or the hormone, the heat shock protein is removed from it, and the DNA binding domain gets exposed, and this DNA binding domain is associated with zinc. Now the next step is, the receptor along with the ligand enters into the nucleus of the cell, and it reaches the enhancer region of the DNA. This region is also known as hormone response element, because it gives response to the hormones that bind with it. This region activates the DNA to undergo transcription, and on transcription it will give rise to a pre-mRNA, and the pre-mRNA undergoes certain changes or modifications, and it will give rise to mRNA. mRNA leaves the nucleus, and in the cytosol translation occurs, and it gives rise to new proteins, and these proteins give response to the cell. So this is how the intracellular receptor action take place. So thanks for watching. I hope you liked it.